what series talks about. So it started with Axel at NCBS wanting to start something to with pollination biology in India, and so there's this under the aegis of umbrella, what's called Indian Pollinator Initiative. Axel at NCBS, Hema Somadathan at ISAT Trivandrum, Vasuki Belawadi at University of Agriculture Sciences, Bangalore, and Professor Pratibha at University of Kolkata have initiated and sort of organized these set of talks. And I'm Joby. I am at University of Hyderabad. I have been mostly just tagging along. Okay, so Christina Grozinger. I'm sure most of the audience here is familiar with her work. She's the distinguished professor at the Penn State University, and she's also the director of Center for Pollinator Research. And probably it is something which you'd like, something like that, which we should be having in India, I would say. And she's a highly accomplished researcher, has carried out a variety of transdisciplinary research, mostly looking at the genomic underpinnings of behavior and physiology surrounding bees and their interacting partners, and which includes prominently even human activity, these including economic downstream consequences of for the bee life and bee and pollination in the ecosystem. Yeah, so without much taking much time, I invite her to deliver the lecture on supporting pollinator biodiversity through transdisciplinary programs. Yeah.
Thank you for that introduction, Joby, and thank you, Axel, and everyone for inviting me. I wish I could be there with you guys in person, but maybe maybe someday. So I have set up. Let's see this work. Oh, I don't think that's working.
Yeah, we can hear. See.
Can you see? What do you see?
Yeah, we can.
Do you see the screen? Okay.
Yeah, full screen.
Okay, perfect.
All right.
Okay, I think this is okay. Great. Okay. So I was going to kind of give you a couple of different stories, kind of related to sort of our efforts to use sort of these transdisciplinary studies where we bring together collaborators who have expertise in different areas to address what we think are kind of the key issues in pollinator health and biodiversity conservation. And I wanted to sort of define sort of what transdisciplinary means to us, just because I think there's a lot of different ways that people sort of think about this. It's not just kind of multidisciplinary or kind of cross-disciplinary. It's a little bit more detailed than that. And so these were some key points that I got from this book, which you guys can get and look at in more detail. But the idea is that you're integrating knowledge across academic disciplines through nonlinear and reflexive interactions. That means not just saying oh, this tool that analytical chemists are using is useful for me, I'll just use it to get this one data point. It's more of a back and forth dialogue and sort of merging and integrating of these fields. It sort of works best if you're sort of in a context-specific situation, so if you're aiming to solve a particular problem and working together to do that, again, so you're not just kind of adding pieces together, but integrating to address some common issue. And these problems are usually sort of driven by sort of real world issues, right? So there has to be communication through the different groups, which also includes practitioners, right from the beginning. So often, what happens is that you sort of know about a general issue in the world and you say, oh, I can study this piece of it and surely that will be helpful. But then when you actually talk to a practitioner who's trying to use that knowledge, it turns out that there's some other element that, other sort of sociological or economic limitation that they face, that they can't really put into practice this thing that you have discovered in your research. So sort of talking to the practitioners and the policymakers sort of early on to understand what they need and what their challenges and limitations are is really important. And then again, it tends to be sort of action oriented, so that you're not just doing sort of theoretical work, but actually trying to, again, think about how this might actually be applied in the real world. So I'm going to have sort of 3 sections to my talk today. The first is focused on our studies looking at nutritional ecology, the second on landscape ecology and the development of decision support tools, and then the third one we'll be talking about sort of what we have thought about and what we have tried to do when we've been building our Center for Pollinator Research. And sort of after each of these sections, I'm going to stop and take questions so that this is a little bit more engaging and a little bit more dynamic, and it's not just me sort of talking at this PowerPoint screen. So for our studies of nutritional ecology, they were sort of motivated because of years of research that we have been doing on understanding the effects of different stressors on bees at the genomic level. And so I'm sure you guys have all seen this before, but we know that bee declines are due to pathogens. You guys can all see my cursor, right? Hopefully, maybe. Anyway, well, I'm moving a cursor around, so you can see it.
Yes, somehow
okay.
It's not. Yeah.
Okay, so there's viruses, there's parasites, which in the case of European honeybees, varroa are a serious issue. There's management practices, which in the U.S., of course, there's like a large-scale commercial pollination service where like 60% of the honeybee colonies are trucked across the country to go to California for almond pollination in February, for example. There's pesticide impacts. There's lack of floral resources, which means lack of nutrition. So because of monocropping and the loss of natural landscapes, the bees don't have as much diversity or as much abundance of flowering plants. And then, of course, there's climate change issues. And so my lab had done work on looking at how bees were responding at the molecular level using transcriptomics approaches to pesticides. And interestingly, we saw that genes associated with sort of nutrition and diet were being affected by pesticides, right? And so this suggested that if you had sort of a more nutritious diet, you would be more resilient to pesticides. And we showed that in one study that was published, and then another one that my current graduate student, McKaylee Crone, is working on. We also looked to see how bees responded to nosema and varroa, and again found that a lot of genes that are involved in metabolism and are also affected by poor or rich diets were affected by these parasites. And then finally, some nice work from Gloria de Grande Hoffman also showed that if you had a pollen-based diet versus an artificial diet, the bees had lower levels of viruses, right? And so overall, what this would suggest is that if we can kind of improve this situation, if we can improve the nutrition available to bees, we might make them more resilient to these other stressors in the landscape and improve health outcomes. So then that led to the question of, if we know that pollen-based nutrition is so important from these studies, then how do we define or identify the best plants that provide the best pollen for bees? Because we know that there's a lot of variation in pollen. And so the next series of studies I'm going to talk about were carried out by Anthony Vaudot, who is a PhD student in my lab, and he's now a postdoc at University of Nevada in Reno. So Anthony made this cute little sort of nutrition fact sheet for pollen, and sort of looked at what was in pollen and what bees basically were using pollen for in their diets. And so bees, of course, required 3 main macronutrients, so fats or lipids, carbohydrates, and protein. Carbohydrates they're mostly getting from nectar, but pollen is really their only source of fat and protein, as well as a number of micronutrients. And we know from studies from Ralston and Kane that protein and lipid concentrations differ dramatically across different plant species. And if you feed bees pollen from different plant species, you get different health outcomes in terms of immune function or development or reproduction. And so Anthony's hypothesis was that when bees were foraging on plants to collect pollen, they were foraging in a way that optimized the ratios of these two key macronutrients, so the proteins and the lipids. And so they were not necessarily trying to maximize either of these, but they're trying to get a balanced ratio. And this is based on the nutritional geometric framework that has been sort of applied and tested in a number of different systems, right? But it had not really been looked at in bees up to this point. So we wanted to look at this, and to do these studies, we started using bumblebees because we needed to have sort of an enclosed arena to have a very controlled experimental system. And so we identified from a previous field study that had been done by Laura Russo when she was a postdoc at Penn State, where she looked at visitation rates of a series of native plants in the field. And so we identified plants that had high, medium, and low visitation rates by Bombus impatiens bumblebees. And then we obtained these plants from growers and we put them into a hoop house in this sort of grid setting, and then put 2 commercial bumblebee colonies into the hoop house with them. And so this was very controlled because the bumblebees could only forage on those plants and nothing else could forage on those plants. Anthony rearranged the grid so that the bumblebees, typically they trap line, and so they just go to the same they follow the same foraging route from day to day, but he rearranged these plants so that they would have to make fresh choices. He controlled the number of bees that were in each colony. He opened the colonies to forage only when all the plants were presenting their pollen so that there was no phenological differences there. And then he tracked the number of bees visiting each individual plant, and he tracked individual bees as they flew around the plants. So again, a very, very controlled study. He then also collected the pollen from these plants by hand and then analyzed its nutritional content. And so what did he find? So these were the series of plants where he was able to get both visitation rates and analyze the carbohydrate, proteins, and lipids. And then he also did the protein to carbohydrate ratio and the protein to lipid ratio. And when he compared this to visitation rates, the carbohydrate, protein, and lipid, and the protein carbohydrate ratios were not significantly associated with visitation rates at all, but the protein lipid ratio was. And so the higher the protein lipid ratio, the more visitation the plants received, right? So the more attractive they were to these bumblebees. So these plants, of course, are full plants. They have all of the floral cues that are associated with having flowers. And so these floral cues might have been influencing the attraction of the bees. And so to look at this in more detail, he did a series of cage studies where these cage bees were offered hand-collected pollen from plants or pollen that had been collected from honeybees and then modified to alter the protein lipid ratio. So it was the same pollen, but then altered nutritional content. And then he also did synthetic liquid diets. And so I think I have this little video here. Does that work? Yes. So you can see this is the pollen being offered in 2 little containers. And so obviously the bees really like this pollen and not so much this one. And so when he did all of these different assessments, he found that the bees preferred diets that had a 5-to-1 to a 10-to-1 protein lipid ratio. And if it was lower than that or higher than that, the diets were not preferred. And so that suggests that the bees are in some way actively selecting for this particular ratio. And so more recently, Anthony published a sort of large-scale analysis where he analyzed the protein lipid ratio from 82 different plant species. This was done in collaboration with many, many co-authors. And so this was sort of accumulating the data from multiple studies. And what you can see is that there's a broad range of protein lipid concentrations that you find in these plant genera. And then he also analyzed the protein lipid ratios that were coming from bee collected pollen. So this was from bumblebees, this is from Osmia orchard bees, and then also from honeybees. And so what you can see is that there's also a spread in terms of what the bees are preferentially collecting. And so theoretically then this would say that, you know, if you're trying to support honeybees in your landscape, then you would maybe plant these particular species. And if you're trying to support Osmia, maybe you would plant these species that have sort of the preferred ratio. But I will say that, you know, at this point, the bee preference studies have been kind of limited in terms of scale and scope, you know, which landscapes these pollen samples were collected from. And so I think we need to do a lot more of this to really understand if you can sort of, you know, design an ideal sort of pollinator planting scheme for specific kinds of bees. But that, I think, is kind of a really interesting area to think about. So we've also been doing a number of studies to try to understand, you know, when bees are foraging, what are the best plants for them? So in addition to the kind of work that Anthony was doing, which is looking at individual selected plant species and then monitoring visitation to those, we also have been using DNA barcoding of pollen as a way to sort of explore what bees are collecting in a particular environment. And so the next study that I'll tell you about was carried out by Doug Sponsler when he was a postdoc in the lab. He's currently a postdoc at University of Würzburg. And so Doug was really interested in studying in urban areas what were honeybees collecting and what was contributing to their colony growth. And so the general question here is that in a particular area, in a particular location, there will be a sort of resource flow that will happen over time, right? So you'll get a lot of flowers blooming, a lot of nectar, a lot of pollen at certain times of year, and then less than other times of year. And then there's different plant communities, of course, that are supporting these different time periods. And so how do we sort of study that? And so the way that Doug studied this was by using these continuous weight monitoring systems. So we can get these automated hive scales. The ones that we used were from a company called Broodminder. And you can put these underneath your colonies and they record the weight changes hourly. And so then Doug spent a long time trying to work out how to take this very complex and very messy data set and turn it into something that we could actually analyze, and we could sort of see, again, changes in overall weight in the colony over time, which pretty much corresponds to changes in the amount of nectar that they're bringing in from the environment. To figure out which plant species were contributing to these resources, he used DNA barcoding of the pollen, and this was using a methodology that was developed by Rodney Richardson when he was a grad student with Reed Johnson. He is now an assistant professor at University of Maryland. And then finally, to be able to do this study in this, you know, complex landscape, we partnered with a beekeeper. This is Don Shump, who had apiaries throughout Philadelphia, which is where we did this study. And so these were a number of rooftop apiaries and then also sort of more standard apiaries, and they covered a broad range of different urban areas. And again, sort of the thought was that, you know, if you were trying to assess what the flowering plants are in one of these areas, it's really challenging because, you know, this is not sort of, you can't easily do a transect and sort of see what the bees are foraging on because there's so much heterogeneity in this environment. So by using these sort of sentinel colonies, you can see what, again, the resource flow is and then what plants the bees are finding in these landscapes. All right, so what did Doug find? So he found that over the 2 years of our study, there was clearly a springtime flow and a fall flow and then a summer dearth. And this sort of pattern has been pretty well documented in sort of temperate systems in the United States and in Europe, where there's sort of a lot of colony growth, a lot of honey production in the spring, and then in the fall. But then in the summer, there's this sort of nutritional dearth period. When he analyzed the pollen that the bees were bringing in and looking at the overall richness of the genera, what he also found was a similar sort of dearth in the summer period and then sort of more in the spring and then more in the fall. And he did an MDS plot to sort of separate out these different pollen types. What you see also is that the community of plants that the bees are harvesting pollen from is really distinct, again, in the springtime, in the summertime, and in the fall. So you're getting sort of a turnover of the plants that the bees are foraging from. And then he analyzed sort of the traits of these plants, looking at what kinds of plants. Are they trees, shrubs, vines, or herbs? Are they perennial or annual? And then are they native or introduced? And basically what you find is that in the springtime, they're harvesting from pollen from native trees and shrubs, and then they move on to these sort of perennial herbaceous plants. And then in the fall, they are going into more vines. So again, I think this is a really powerful way to sort of assess what the bees are preferring in a particular landscape without having to do sort of very, very intensive field studies. And again, like some of these things, like for the trees, it's very hard to do visual observations. And so using this barcoding analysis, we can sort of see these trees very clearly in our analysis. So at this point, I was going to stop and take questions. I can stop sharing so I can actually see you guys, maybe. So if you guys have any questions, I would be happy to answer them. Yes, I think you should just yell out. I'm having sort of some issues with my screen. Is this letting me go? Yeah, so just, I can't hear you. I see somebody raise their hand, but I think, can you just unmute yourself and ask.
Yeah, it's nice listening, Christina. So myself, Pradeep Chuneja. I'm head of Department of Entomology from Punjab Agricultural University. So in fact, you have mentioned a new kind of dimension in respect of the bees' preferences for the different bee flora. So you mentioned the ratio of the L and P, L is to P. Yeah. So I was looking at that. You were talking about the three bee species, and I could recollect when I did my doctorate during '91, and I was recording the data. I have recorded the data during '86 to '90, and the average protein content, that is accrued protein content in our pollen, that came out to be around 19.72, and the lipper content was around 8. So that way, it is around 2.5. So I was looking at that. It is just matching your data, which you have pointing out. So we didn't think from this angle of from the LNP. We had a different objective. In fact, we had worked out the nutritional status of our colonies with respect to the availability of the bee flora. And we try to develop certain diets, some pollen substitute and supplements that they should be equal in respect of their chemical composition to the available bee flora. Yeah. So it is just, I was going to share that it is matching for the mellifera. We have all the mellifera. We want Onapis mellifera. Yeah.
Yes. No, that
and, really yes. And the other information about the bee flora in the region. So that's very important for planning the migration, because this melliferous beekeeping that can't do without migration, it being a very voracious feed. Yes, because the floral availability that varies in time and space. So this information in the state and around. So that's very important in planning the migration or guiding the beekeepers for enhancing the productivity, the colony productivity.
Yeah, I would
That's
love
it.
to see maybe a copy of your paper if you publish that. That would be really interesting because I mean,
Okay. So it's a
yeah,
long back. We had some certain publications in Indian Bee Journal during 1991, 92.
yeah.
So yeah, it
Well,
is a long back, in fact.
yeah. If you could find a copy, that'd be interesting. I mean, we've been doing some follow-up work with honeybees, and they seem, but again, I think we need to look at this in more detail. They seem like they collect a lot of things, but then on average, like it averages out to this this ratio. And so that, that becomes very interesting because you think like, are they just collecting what's available? And so then if you don't have a broad enough range of things available, they'll be using, as their food source something that's not as nutritious. But I think there's a lot of, I think there's a lot of interesting questions there.
Yeah, then this nutrition, that's very important, nutrition of the bees. So in fact, we didn't take the crude protein as the parameter. We took the true protein that came out to be 14.72, and we had another parameter, that this crude fiber. We kept it below the average which we got in the natural pollen, and we also then tried to make it more nutritive through the addition of the skimmed milk powder. But the Souls and co-workers, they had already reported in 1961 that the lactose that is toxic to the honeybees. So we shouldn't have it in the diet more than 2%. So because our this skimmed milk powder that had 51% lactose, so we kept its proportion as 4%, so that we shouldn't have the lactose more than 2%, and we are able to add up the nutrition in terms of the protein of the diet. So
right.
This was
Yes, no, and I think this is something that, that the beekeepers and the beekeeping industry is so interested in, is sort of like, how do you make kind of an ideal diet? And I think for the most part, the lipids have not been considered very much with those diets. It tends to be sort of like, how do we kind of maximize the protein? And there is some work coming from Sarah Leonard's lab that would suggest that having too much lipid is also not good for the bees. So again, it's like, sort of getting the right ratios so that they have what they need, but they're not overeating on anything. So
the ratio you had been pointing out, that is going in favor of the protein, because you are mentioning it is P ratio L, 4 to 5. But there have been reports that if in the honeybee diet, if we have the protein content more than 23%, that causes the toxicity in the bees.
Right. Yeah. So it's all, I think,
that's also there.
I think we'll have to think about a more, nuanced way of trying to understand some of this. And then also seeing, I have a postdoc in my lab, Gabriella Quinlan, is looking at sort of seasonal variations. So like when bees are in different parts of their colony cycle, do they have different nutritional needs, right? So
definitely. Even here is the because I hear from Punjab, that's the northwestern state of the country of India. Here during the fall, we don't have much of the flora, so there is a dwindling of the colonies. So but I could see that the brood rearing was actually matching with the availability of the pollen.
Yes.
So we had the brood rearing at its ebb during the monsoon season during the fall.
No, I think it would be so interesting to look at, to again look at like a tropical system, and then also, looking at some of these other species to see sort of how they approach this sort of nutritional balancing.
So okay, thank you.
Are there any other questions, or shall I move on to my next section? Oh, Joby, I think you're muted. I don't know. I can't hear you. Oh, sorry. I'm not sure. I guess I will keep moving on. But if there are any other questions, so it should be time at the end. Okay. All right. So hopefully back now. Of course, you guys have disappeared completely now. I love this sharing. All right, well, I'm going to hope that everything is okay. Okay, no, you're in the back. Okay. All right, so next I'm going to talk about sort of our studies of landscape ecology and using this to develop decision support tools. So all, our work on plants is great, but, that sort of assumes that you have some control over the plantings in your area, which, typically you can't plant out enough plants to sort of support your bees in every place that they might be foraging, because bees can forage over several kilometers. And so we wanted to sort of ask this question of, like, how can we help people know generally if they're in a landscape that will be good for their bees without having to do sort of these detailed studies like what Doug did, or without having to do detailed transects, for example. And so the project that we're using to address these questions is called BEESCAPE, and this is a collaboration between multiple people and multiple institutions, and it's supported by a number of different sources. And basically what we want is that if you're in a landscape like this is like sort of a pretty typical sort of agricultural landscape in southeastern Pennsylvania. Your bee colony might be in the middle here, but your bees can be foraging over this entire area, right? Which is covering a lot of different crops, so different land use patterns. And so we wanted to have some way that people could understand sort of what the floral resources were in the spring, summer, and fall. For wild bees, what the nesting habitat options are for bees, and then what sort of insecticide exposure and risk they might be dealing with. And so to do this, we developed a couple of different indices of these different traits, I guess, for the landscape. So the first two were developed by Eric Lonsdorf, actually, several years ago.
Christina,
oh,
sorry,
yeah.
your screen is not visible.
Oh, my screen is not visible?
Yes.
Okay. Thank you. All right, let me try this. Does that work? Do you see it?
Yeah, it's coming up. Yeah, no. Yeah, it's visible. Yes.
Now it's full screen? Okay, thank you. Sorry.
Yeah, cool. Yeah.
Is that okay? Okay, you see what do my bees experience?
Yes, we do see that, yes.
Okay, all right. Okay, just yell out, because like I said, somehow I can't see you guys when I got my screen up. So if you have any issues, just yell. Okay, so sorry. So this was BEESCAPE. These are all our people that we're working with. And so again, this is our agricultural landscape, and then these are the three different indices that we're interested in sort of mapping out and then having accessible to people. So the Floral and Nesting Resource Index was again developed by Eric Lonsdorf, who's at the University of Minnesota in the Natural Capital Project. And he developed this a couple of years ago. And the basic premise behind this is that, we have these land cover maps of the United States that have all these different classifications of land cover and land use. And so typically you would sort of say in the area around, my site, I have this % agricultural land, or this % natural land, or this % corn. But it's, that is very specific to your location. And so it makes it hard to really, take that information that you get and translate it into other different kinds of locations. So Eric's index basically takes these land cover classes and asks experts, which would be experts in the field of pollination or pollinator biology and conservation, sort of how they would rank these different land cover classes in terms of floral resources. And again, so the experts then provide their input. This gets averaged, and so then each of these land use categories gets a particular score. And so then when you go back into this landscape, you can essentially get scores for all of these locations that sum up the different kinds of land cover classes that are present at that location. So instead of having all of these different classes, you can get this sort of smooth scoring system that goes from 0 to 1 for any particular location, right? And so then if you know that, for example, your bees can forage over a 3-kilometer radius, you can sort of average the scores for that 3-kilometer radius, or use a distance weighting function so that the scores that are closest to your site are weighted more highly than scores that are further away. And so again, Eric did this for both floral and nesting resources. For insecticide toxic load, it was a more challenging computational question, and this was work that was done by Maggie Douglas, which she started when she was a postdoc in my lab and then wrapped up when she's now an assistant professor at Dickinson College. And basically what we wanted was not just information about sort of the amount of a particular insecticide or pesticide that was applied to the landscape, but how this could impact bees. And kind of aggregating across all of the insecticides that were used. And so the ultimate score that we wanted was this insect toxic load, which was basically, given what we expect for the types and amounts of insecticides applied to a particular location, how many bees would die on that acre if all those drops of insecticide actually came in contact with the bees. So this has nothing to do with exposure. This is just mapping out exactly what is being put on the landscape and assuming all of it goes on to bees. And so to do this, we got information from the USGS, which has pesticide use data for different crops across the different states. So we know how much is applied. And then we can map out the crop area in a particular location using the cropland data layer. And then using data from the EPA and Ecotox, we could get the toxicity scores for each of these insecticides for bees. And so again, aggregating across all of the insecticides that are being used in a particular location, we can create this insecticide toxic load score. And so that means then by using the cropland data layer, we can map out these different indices at a 30-meter resolution across the United States. So we did this for forage quality, for example. We did it for nesting quality, which I'm not showing a map of. We did it for insecticide toxic load, which is over here. And then we also did it for the economic value of pollination services, which is shown down here. This is a county level map. And this was done in collaboration with Vikas Khanna, who's at University of Pittsburgh, and his two students, Alex Jordan and Mason Unger. The economic value of pollination services is not part of BEESCAPE yet, but we're hoping to include it sometime in the future. But the forage, nesting, and insecticide toxic load are. And so again, with BEESCAPE, what we wanted to do is create a web-based decision support tool where a user could basically go in and find their site, drop a pin on it, and get scores for seasonal forage quality, nesting quality, insecticide toxic load, and then at different distances from their site. And then over, I think in May, we made this available for the entire continental US. So just to kind of show you how this would work. So this is the map of State College, Pennsylvania, which is where I am. And so you can drop a pin on this, and then it pulls up the scores for nesting quality, insecticide toxic load, spring, summer, and fall floral quality. And these scores don't really mean a lot without comparisons to other things, but you can, if you hover over this in our website, it tells you whether this is considered a low, medium, or high score. And there's also an option to add the crop data layer over it so you can see what crops are being and other land use categories are happening in your area. So just to give you an example of a comparison, so if we drop a pin outside of Urbana, Illinois, which is where Axel and I were postdocs, this is a much more agricultural area. And so if you compare the scores, you can see that the nesting scores are much lower, the insecticide scores are much higher. And then also the floral resource scores are a lot lower. So this would suggest overall, State College in this particular location should be a better area for bees because there's more resources and less risk than what you would find in Urbana. But of course, this is all at this point, this is just mapping out these indices. And so there's not really a connection with actual bee health. And so we've been starting to do the studies to try to see how well these indices match with bee health, and then we can hopefully integrate across them so that instead of just saying, we think that floral resources will be low here, we can say overall we predict, this outcome for your beehive at this location. And so we have a study looking at wild bee diversity and abundance that was done by Melanie Kammerer, she was a grad student in the lab. And we have a study on bumblebee pathogen and parasite loads, which is carried out by DJ McNeil in collaboration with Heather Hines. And just to give you an example of what that looks like, so this is looking at the disease index. So this is all the pathogens and parasites that we found and sort of aggregating them, and then looking to see how this disease index changed in the bees that we collected from across Pennsylvania according to these different traits, right? So this is spring floral resources. So that's that floral index. And you can see that as the resources improve, the disease index decreases. As the nesting habitat quality improves, the disease index decreases. So again, more resources, less disease. If you have more honeybee colonies in the area, you actually get an increase in the disease index, which we know from other studies that honeybees can transmit many of these pathogens to the wild bees. And we're also seeing an effect of sort of latitude and longitude, which we still have to sort of work out. But based on some of our other studies, this is probably something to do with microclimate. We also looked at honeybee winter survival. And so this was work that was done by Martina Kalovi, who's a postdoc with Sarah Gosley, who's at the USDA. And for this, we got data from the Pennsylvania State beekeepers because they do a survey every winter to see survival of colonies. And so they very kindly added in information where people could share with us their GIS locations of their apiaries. And so this was a large data set of over 1,700 colonies in 3 years. And so when we did this analysis, we found that it was not the landscape indices that were predictive, but rather weather conditions from the previous growing season, which is, you know, kind of disappointing, but not super surprising because the beekeepers are, of course, like feeding their bees and managing the parasites. And so there's a lot of management that goes on here for kind of nutrition and pathogens, but you can't really manage the weather. And so just to give you an example of how we use this information then to, again, try to create a decision support tool that could help beekeepers. We made this Bee Winter Wise decision support tool where, again, a beekeeper can drop a pin at their location and then they can sort of see the growing degree day accumulation over time and see the model prediction of, you know, this range would be sort of too hot for the bees, this range would be too cool, and so then they can see where their site is in a particular year so that they can sort of make some plans for that. So I'm going to take a break again and see if there are any questions about this. And you guys were able to see that okay, I hope? Yeah. You guys have any questions?
I don't see any raised hands so far. Yeah, one raised hand. Dr. Pradeep. Yeah, I've unmuted you.
Okay, Christina. So it is nice to listen that the diversity and abundance that is being mapped against the presence of any pathogen or the parasite and also the pesticide use. But there is another one parameter that may also be included, that's the air quality.
Oh, yes.
Yeah. So concentration of the increasing noxious gases like NO2 or methane or increasing carbon dioxide concentration. So they are also impacting, and even the increasing sound pollution because of increasing industrialization and urbanization. So these are the parameters which these are the parameters which also need to be considered while looking at the diversity and abundance, because these we have observed here that these are also impacting.
Yes. Yeah. No. And we have there's a collaborator of ours at Penn State, Jose Fuentes, who's done work on how pollutants interfere with pollinators' ability to find flowers, right? So we've talked to him a little bit about trying to have kind of like an air quality index. But if you have some suggestions for sort of what would be the key parameters to think about for that, that would be, I would definitely be interested in talking to you about that a little bit more. Sort of like, yeah, I mean, there's many aspects of air quality. So what do you know, which would be the most important to consider? And again, it has to be something that we can kind of easily pack into an index, right? Into something kind of simple that gets aggregated. So yeah, so maybe afterwards we can have a talk about it.
Particularly from the Indian point of view, so this is very important because the land holding here in the country, that's much smaller than in the states, and the colonies the farmers they are keeping, so they are along the roadside or very close to the industries or roads, so they're not though it's in the open landscape, but it's not far away from these kind of the situations. So, yeah.
And I know
all these.
Eric Lonsdorf, for some work they've been doing on monarch conservation, I know they have included roads because you know there's often a lot of forage along roads for monarchs, but then the monarchs they also get hit by the cars. So I think they've made some models that are related to like distance from roads and then also the speed that people travel. So maybe if you know if most of the pollution is coming from roads, there might be a way to kind of again sort of model that in a way that's pretty generalizable.
So near to the road, so because of the increasing vehicular traffic and this pollution because of these exhaust gases from these vehicles. So all these they have.
I think you know, Axel has a question. Axel, yeah.
I actually have a question not to Christina, but to my Indian colleagues. I mean, I was really stunned by the data set that you have and that you can kind of plot it on. And I would like to know whether actually such data sets are available for India too. I mean, do you know, I mean, I guess that Europe, they would also probably have a lot of these data. And
I think surprisingly, Europe does not have a lot of data on the pesticides, which is very interesting. I mean, so yeah, but anyway, so yeah, I'd be curious to know also like what information you would have available.
Yeah, so does anyone know? Dr. Avinash, do you know whether we have data for India in that? Someone has to unmute him.
Yeah, no, you are unmuted. Excellent.
No, Dr. Avinash.
Yeah, I can hear you.
No. I asked Dr. Avinash whether he knows whether there are.
Oh, just a minute. I think.
Axel, what data you have been asking for?
No, kind of like these data that Christina has. I mean, pesticide use, floral abundance, right? Whether we have maps in India at that same detail, or at least we have maps.
Yes, we have the data, the state-wise, the pesticide consumption data we are having. How much is the quantity of the insecticide that is being used? How much is the herbicide and how much is the fungicides? So we have the complete data. So
is that available?
All this information that's available at the website of our Directorate of Plant Protection, Quarantine and Storage.
Okay, good. Thanks.
Yes, and if you need that data, I can provide you all the data. In India, this average pesticide consumption that is around 320 gram per hectare.
But
that's for the country. And the maximum consumption we have in the Maharashtra, followed by in the Uttar Pradesh. Then third is the Punjab. And if we talk about this per unit area-wise consumption, in that case, our northernmost state, that's the Jammu and Kashmir, that stands at number 1. And, followed by then Tripura, then is the Punjab.
But then these data are probably not digitized, or are they digitized? I mean, the data, or are they in table?
Yeah, that is in the table form. We can provide you. No issue. I'm having all the data. Yeah, up to the last year. Yeah.
Okay, thanks.
Yeah.
So just to follow up on that, so is the data for each crop, or is it like on a county scale?
Yes, so far as the Punjab, my state is concerned, we have the season-wise data. But for the other states, we have the annual data.
Well, I can send you a copy of the paper that Maggie wrote, so you can kind of see what she did. But if you have sort of the crop-specific pesticide data, and then if you have some kind of land cover, data set that you can then sort of merge with those, and you can basically recreate the same kind of thing. And it's, for us, I didn't tell you all of the analysis that we've done, but because we have data going back for like 20 years, we could actually sort of see how insecticide use patterns were changing over time, and sort of see areas where the landscape was becoming more toxic to bees, and like what crops were contributing to that. So even just sort of the mapping process itself, without worrying about making a decision support tool, can tell you information about, like how are things changing for bees? Like, is it getting better? Is it getting worse? Which areas where it might be of concern? So I think it's a valuable process, I guess.
But because of the increasing this environmental and health concerns, so there is a lot of awareness now, and the pesticide use that is coming down continuously, but particularly in the states where which were having the maximum use. So there is a considerable reduction over the past couple of years.
I will say also that in our case, there's been a reduction in the amount of pesticide, but the pesticides that are being used are more toxic. So even though like the quantity was decreasing, when you calculate the toxicity was increasing. So that was another sort of interesting thing, because there were different things that you heard from people, and so the quantity itself is maybe not as informative as the toxic load.
So, in fact, so far as the toxicity is concerned, because here our CIBRC system, that is a bit different from the toxicity classification that has been given by the WHO, but it is almost overlapping. And here now we are at the verge of eliminating all the red chemistry molecules from the country. And if I talk about the Punjab state, which is the agrarian state of the country, which with the maximum crop productivity, so here only one insecticide that is red chemistry that's left. That's a carbofuran. So that's a carbamate insecticide. So otherwise we have two rodenticides, aluminum phosphide and bromodiolone. So these are coming from the red chemistry. Otherwise we have these only three molecules left. And we are also phasing out the yellow chemistry molecules, and now we have moved towards the blue and green chemistry molecules. And also the alternate control measure, pest control measures. Yeah.
So I think, Axel, did you have something?
That's leaving now.
Yeah.
I'm not sure if Axel had something you wanted to add.
He's no more.
So yes, that Punjab be different from other states. Actually, there's also a question. I mean, agriculture is based on states. Agriculture is based on that really knows what small farmers put on pesticides on their land, and it's probably better observable if you have big growing areas of apple kind of plantage that you have in Punjab, where you have a big grower who is only and who is basically kind of like also writing it down what he is using. But in many, so from all what I know, but I'm not an expert on it, I think it's probably very diverse and also maybe not really recollectable what people put on land, and then even probably you cannot kind of measure the soil and kind of see what has been done to it. So what to say? Other colleagues have to kind of tell. I'm not an agriculturalist.
Yeah, in the Punjab, the productivity has gone only for the cereal crops, only two. If you take wheat and rice, that has gone beyond 12 tons per hectare, more than 12 tons, only for the two crops. And you have, yes, and that is the highest in the world. That's the highest anywhere in the world. And regarding the apple, we don't have the apple. Apple is there in the Jammu and Kashmir and Himachal Pradesh, but yes, yeah. So there is a lot of per hectare. They are using 830 gram, Jammu and Kashmir people per hectare. So per unit area, their consumption is very high, and maximum of the pesticide application, that's on the apple because of kind of the diseases.
Well, I think there's also, you know, a benefit of, you know, like if you have a goal to make some index like this, to be able to say, okay, well, where are the gaps, you know, and in our knowledge before we could make this, and that can also be, I think, a very useful, again, exercise. Like in our case, you know, what we found was that the main contributor to the toxic load was the seed treatments on field crops, and the USDS stopped gathering data on those seed treatments for a couple of years, and now I think USDA is picking it up. But there's like this gap, right? And so, you know, understanding that, well, we actually have no data on this, you know, I think is again just valuable to kind of know about. But maybe I should keep going so that we don't keep you guys too late on a Friday.
But Christina, in spite of the Punjab having the third largest user of the pesticides, it is number one in beekeeping, and we have only exclusively the melliferous beekeeping. There is a number one state,
right?
In respect of the number of the beekeepers, number of honeybee colonies, then honey production and export. In fact, the melliferous beekeeping in the country that took off from the Punjab in 1976 with the release of Apis mellifera into the country by the Punjab Agricultural University. Yes, Christina, yes.
I think
your job
we
is
can take the questions in the in the end.
I can understand.
I will try to keep going, and if you could tell me whether or not you can see my screen.
No, now we.
You see it? Is it big now?
Yeah, now we can. Yeah, but
okay.
Now it's big. Yeah.
Okay. So now I'm going to talk about sort of my third topic, which is sort of what our approaches and goals were with building our Center for Pollinator Research. So here's our logo. So we are supported by the Penn State College of Agricultural Sciences and the Huck Institute of Life Sciences. This is our website. If you just look up Center for Pollinator Research Penn State, you should find us. This is our mission statement. So we're committed to developing and implementing integrative multidisciplinary approaches to improving pollinator health, conservation, and management for ecosystem services through research, education, outreach, and policy. So that's a lot of mouthful, and it's pretty standard. But really the way that we were thinking about this was we wanted to do less work and have more impact. And because we have more impact, we can have more resources and do less work. So that was sort of like the underlying goal was to sort of, you know, create this collaborative group that could be more efficient and more effective. And so our sort of priorities were to be recognized as a world leader in pollinator research, improve the quality of our research, education, and outreach efforts, improve the efficiency of our efforts, and then facilitate collaboration among our center members, and then also with the broader community, including collaborators outside of Penn State. And so we wanted to, you know, to create this framework or ecosystems that allow people with diverse interests and experiences to contribute and thrive. And so when I think about this, you know, I think a framework is like if you have a very specific problem that you're trying to solve, you know, and so everyone is again working towards that particular goal. Versus an ecosystem is more where, you know, there could be new and emerging problems, but we have a group that's already kind of working together and is sustainable kind of over time. And then that group can kind of be repurposed in different ways, I guess, or to form different kind of configurations to deal with different problems. So we really want kind of more of an ecosystem that will kind of persist across time and across different problems and collaborations. And this is, I have to actually update this. So this was our group from about 1.5 years ago. We've added some new people. Some folks have retired. But we have a really large group that spans, I think, 8 different colleges at Penn State. And obviously, you know, sort of the most active people are within this Department of Entomology, but we have collaborators who are coming from the plant sciences, from engineering, from education, from computer science, from biology. And so again, very broad, and all sort of united in this idea of, you know, how can we use our expertise to support pollinator research and conservation. I think a really important thing is kind of silly, but we made a website, right? So this website became sort of the central unit for kind of organizing information that we were trying to get out, to talk about sort of our research outputs. And I think it's been really successful. We get over 100,000 views a year. We have a number of news articles that we post, and these news articles are, you know, press releases coming from Penn State, but also when people interview us from other organizations, we can sort of put a link to that article or that interview on our website. And actually, like, a lot of the information we're aggregating is coming from outside of Penn State. It's not being generated by us. There's a lot of different kinds of information. So we have a lot of information just for the public. We have information for other researchers. We have a pollinator listserv, which is, there's one that's within Penn State and then one that is regional. And the regional one is, again, very active, and it's a way for people to communicate information about new research findings or new positions. And then we sort of take all of this information and turn it into a newsletter that we then sort of post. We have now a weekly newsletter, and then we also have one that sort of aggregates everything over the year. So sort of different ways to try to get this information to people. We, as our big sort of kickoff event, we created this conference, this international conference on pollinator biology, health, and policy. So the first one was in 2010. Our center was formed in 2009. And the idea here was that there was sort of a gap in sort of the community of conferences, right? And there's a lot of conferences, and so you don't want to sort of create conferences just for fun, but in this case, we felt that there was a disconnect between people who worked on honeybees and people who worked on wild bees, and people who were researchers versus practitioners, and so we wanted to have a conference that could very nicely bring all of these folks together. And so this conference is run every 3 years. We have about 200 to 250 participants, about at least 15 countries represented each time. And then again, people coming from all sorts of different institutions. And so our goal with this was it's very integrated. So there's a lot of sessions, different kinds of sessions, but there's one session at a time. So everyone has to listen to each other's talks and learn something that maybe is outside of their field. We like to showcase Penn State. So when we host it, we of course have presentations from our own group, but we have, things in a really nice location. We have really nice food, so everyone has a good time. And then we also are using this as a way to connect Penn State with the broader community. So we have new organizers for the conference, and then also for the symposia sessions each time, so that it remains fresh and we're always bringing in outside perspectives. So UC Davis hosted the conference in 2019, and so we think this is probably going to be the model that we follow, is where it'll be hosted at Penn State in one iteration and then move out to another location in subsequent for the next cycle. And again, this was really successful, and this allowed us to sort of bring in people who are from the West Coast as well. We also have a stakeholder advisory board, which is both regional and national groups that we communicate with, and a lot of these folks have become partners for research projects and education projects. And then we established a couple of years ago an integrative pollinator ecology graduate training program. And so it's funded initially by Penn State's College of Ag's Strategic Networking Initiative, and then we receive funding from USDA. And students are coming from all sorts of different programs to be part of this. So this is sort of a cross-department and again, cross-college program. And so the goal with this was, again, to be transdisciplinary. So we were bridging fields. We have a lot of faculty. We started out with 9, but we've added on additional faculty, again, to take into account that project ideas and challenges are sort of changing over time. And so bringing in people with different expertise made sense. We have 9 co-mentored students. They are kind of doing their independent projects, but then they come together for an annual colloquium every year, which ends up being really successful because we also attract other students, faculty, and postdocs. This year, we're actually running Global Perspectives in Integrated Pest and Pollinator Management. It's going to be an online course, so I can send you information about that as well. I think timing-wise, it will not be a good time for you guys to join in because it'll be sort of in the middle of the night for you. But we have, again, sort of a number of really interesting speakers and topics, so I can send that information as well. And then for outreach, you know, we are a land grant university, and so a main point of what we're trying to do is take our information and bring that out, not just to stakeholders, but also to the public. And so by forming the center, it allowed us to consolidate and coordinate our outreach activities. We developed relationships with educators so that it's not just us trying to figure out what would be most effective, but people who actually study the best way to educate and have impact. And again, we were able to obtain funding from a number of different sources. And this is just an example of some of the projects that we have done that has been funded by these different groups. And so once you sort of build something that people are very excited about, then other people will just sort of join in with you to sort of, you know, promote this vision of helping pollinators. And that has been one of the most gratifying things, I think, about creating the center. So for example, a few years ago, there was a group of students in a computer science class that decided that they wanted to make a pollinator video game. So that was awesome. So they sort of used their talents to make this game that sort of showed plant pollinator networks and sort of collapse of pollinator communities and how to deal with that. Our arboretum was started, I feel like maybe about 15 years ago. Then it had a very small pollinator garden. These little circles here were the pollinator garden. But because of all the excitement and interest in pollinators, there's a plan now to expand this to a 4-acre garden that should be done sometime next year. And so there's going to be, I think, about 400 different kinds of plant species and cultivars that will be part of this, and we're going to showcase different kinds of landscapes. So it's going to be a fabulous place for research and outreach. And then again, sort of the graduating class last year, without us being involved, they chose to provide support for creating a gate for this garden. And again, this, you know, they did this without us suggesting it. They sort of came up with this on their own based on, again, sort of the information that we've been able to get out to Penn State and the larger community. And then we've also been able to get donors who are interested in supporting our research and education to endow various programs. So we have an endowed undergraduate scholarship. We had an endowed graduate fellowship, which was then converted into an early career professorship. We have an endowed undergraduate and graduate summer research program, which we've made broad. So it's not just biological or ecological research. It also includes art and humanities. So this was a project from a few years ago where it was a sort of video installation looking at sort of pollinators from sort of a human perspective. And then also most recently, we have an endowed professorship. So I'm now the recipient of this endowed professorship, and it's just really fantastic because it allows you to try sort of new research projects and sort of get those things going before you're trying to apply for funding for these things, which is usually, you know, it's helpful to have sort of preliminary data for this stuff. So with that, I think that's all I've got, and I am happy to take any questions that you might have about this last part or anything else.
I see Sainu has a raised hand. Sainu has to be unmuted.
Hi, Christina. I am Sinu from India. This is regarding your nutritional nutrition physiology part. bees also forage on the microbes in the pollen and the nectar. That is the recent research says. So what is your say on that? You are looking on the lipids and the protein proportion. The one good part about that, the microbes which they forage on.
Yes. So foraging ecology is very complicated, right? So it's not just the microbes, but it's also the micronutrients. And so we took a very simplistic and broad approach just because that was sort of what we were most interested in, were these macronutrients. And so we did not really consider the microbial communities at all. But I think there's a whole interesting additional multiple layers, I think, in terms of the micronutrients, or exactly which fatty acid is present, secondary plant compounds which may have some toxicity but also may have medicinal things. So I think there's a whole layer of things to look at. And again, we partly did our analysis as we did it in this very sort of high level and simplistic way because it was very easy to do on a lot of plants, right? And so that was kind of the motivation for working at that level versus doing something more detailed.
Thank you, Christina.
I do not see any raised hands now.
It was a lot of information, and I'll send you more by an email that you can distribute to people if you like for some of these studies that I didn't have time to talk about.
Yeah.
But I'm happy if there's any other questions.
So I have a question. Yeah, I think Pradeep, Dr. Pradeep has hands up. Can you go ahead?
In fact, Christina, this is in respect of the just previous question by one of our friends. So because from the plants, the bees, they would be collecting these microbes. Those would be they could be the plant pathogens or some symbionts. So I don't see that if they would be affecting the bees' health in any way.
I think that's, again, it's kind of an open area of study, right? It's sort of like, are the microbes helping with digestion or releasing some nutrients? And again, there's other things in terms of pathogen transmission, which we know happens through flowers as well, but whether or not these are using that information to decide what to forage on or not, versus that, the most attractive flower just gets the most pathogens because everybody's visiting it. So I think it's still an open and interesting question, but a complex question. So it's too complicated.
But plants, they are infected with so many kind of the bacterial and fungal diseases. So these diseases are very common. But the bees, they have evolved to survive on the pollen.
Mm-hmm.
So the plant pathogens, because these are not the animal pathogens, because the bees are infected with some different diseases. And we look at the spectrum of the microbes which are associated with the bees and are inflicting or causing the diseases. Those are entirely different from what we get on the plants.
Yeah, so we did do a metagenomic analysis of viral communities, which actually ACTFL was part of that study, where we were looking at all the viruses that were present in bee communities that were collected in different places across the world. I mean, it's pretty clear that the bees do share a lot of these viruses, but again, I think it's an open question of...
But there's not even a single report.
For the bees. Yeah,
there's not even a single report of this crisscross between the plants and these bees for these viruses. So,
there's been a couple.
Because there is a great extent of the specificity amongst the viruses.
Mm-hmm.
So plant viruses, they are not causing any disease on the bees, and the bees viruses, yes, we have an array of 12 viruses. They are well known to cause the disease in one or the other part of the country. But they are not causing any plant diseases.
Mm-hmm.
Because,
Can I add to that maybe? So just sort of naive question, but how the gut flora of the bees are sort of acquired from their other bees or from other sources? I mean, I'm assuming.
From what I understand, I think that there's to some extent sort of transmission within the colony for honeybees, but there are some microbes that are just sort of present in the environment, and so they pick it up through the pollen. And again, I think, you know, I'm not a microbial or microbiome person, but it seems that there's definitely key parts that are very important for nutrition, but then other ones that seem to be more just kind of you know, present in the background and not necessarily having a negative effect. It's a whole, you know, like it's a whole other ecological community in there. So I think there's a lot of different, you know, different outcomes that will happen as we sort of poke and prod that system.
So any other, I don't see any raised hands, so I had to assume that we are sort of. So I would like to thank everybody who came in to listen and participate, and a very special thanks to Professor Christina. So I think we will close the meeting if nobody else has anything to say.
Well, thank you everyone for inviting me. I really appreciate the chance we get to talk to you, and hopefully we can find some collaborations to do in the future. It'd be great.
Yeah.